Hello, welcome ladies and gentlemen to today's tutorial. Today we are going to look at the male reproductive system. You've already seen that of the female reproductive system. Yes, so if you have not gone to watch that video, please do so that you get a complete picture regarding the reproductive systems in humans. Just to start with, yes, I want to tell you something which is a common knowledge. We know that generally males will actually die earlier, than females. And yes, possibly, yes, it could be that yes, males are likely going to take, greater risk, yes, in life. Apart from that, even they want to engage in more dangerous, employments or jobs, such as in even the military. Yes, they are going to be there in the fire service. They are going to be there. Working in some dangerous areas such as mining sites. So it tells you that males are likely to even die early than that of females. Yes, of course, males are also, going to die early, perhaps, yes, because, they are likely to commit suicide. And the reason is that when they have psychological problems, yeah, they don't, seek advice. They think they're on their own. Unlike female who always, resort to, some kind of emotional assistance, that of males tend to be strong and don't want to, engage in that. Even yes, stressing on that, when even males are sick, they don't want to even visit the hospital. Don't want to go to the hospital. Neither do they want to even take in some medication. They think they are stronger, than that of the females. So these are, I think, some of the reasons why, of course, males are likely to even die earlier than females. Yes, even apart from this, looking at how females often use the social media, it tells us that in fact females are quite emotionally, stronger because they have some, social attachments, by way of visiting the social media and then freeing their mind regarding, thinking a lot. So that females, are less likely to commit suicide, as I've already indicated. Now, the striking thing too that we observe is that males are always at the giving end, and females will often receive from males. This one may even be a reason why males are even likely to die earlier. Well, of course, when during sexual intercourse, males are going to ejaculate into the female. Always female being at the receiving end, and therefore, of course, likely to thrive, better in the this planet compared to that of males. So without much ado, let's actually go through this male reproductive system. And remember that the male reproductive system will have gonad being the male main organ or the primary organ of the reproductive system, being the testis or the testicle. And this testicle will hang in the scrotal sac. We're actually going to see how it descends into the scrotal sac, and then to also have some accessory structures which will be secondary structures like the penis, then the intratesticular and of course extracellular duct systems. We are going to see all these ones. But of course, we know that generally, yes, the reproductive system is going to help, produce gametes. Yes, in this case, male gametes. It will help, nourish these gametes. Yes, of course, will help transport these gametes and eventually provide that suitable environment so that after fertilization, the zygote that will be formed will be, get a place to get implanted in the uterus. So let's actually go through this, male reproductive system briefly. So as I indicated, I'm going to begin with the gonads being the testes. So remember that yes, the testes. Generally, if you look at the embryo prior to about the fifth week of intrauterine life, that of the male and that of female are indistinguishable. All right, they are indistinguishable. But what we are going to see is that I mean the testis begins development from the medulla of primordial gonad, okay, medulla of primordial gonad, such that the testis will actually develop near where we have the kidney, okay. So you can see over here, okay, so there's the kidney and here is the testis. So developmentally it grows near where we are going to have the kidney. So what will happen is that the kidney actually ascends, okay, the kidney moves up while the testis moves down, okay. So that what will happen is that the testis what will happen is that in the abdominal area will begin to descend, okay, towards the groin area, okay, towards the groin area, so that we expect that around the 8th week of intrauterine life, the kidney will reach its normal adult position, okay, all the way from actually T11 L to even L2, level. There could be that variation between the left and right kidneys. But of course, around the 7th month, yes, the testes, okay, will descend, and then with the help of what we call the gubernaculum, okay, gubernaculum, eventually it will get to where its final destination being the scrotal sac, sorry, it will go through what we call the inguinal canal, so that just about around 4 to 6 weeks prior to uh, parturition, okay, or prior to delivery, then what will happen is that it should have reached its final destination inside what we call the testis, inside the inside the scrotal sac, So what we are saying is that the testis is going to start development from the abdomen, near where we have the kidney. The kidney will ascend, go cranially, where the testis will go caudally, and then eventually it has to be inside the scrotal sac, So that's what will happen. So what will happen is that what is going to pull it into the scrotal sac will be by what we call the gubernaculum. Gubernaculum is going to do that. Now this gubernaculum, yes, you can also call it the scroto-inguinal ligament, or better still, you can call it the posterior gonadal ligament. This is the gubernaculum. But usually what will happen is that as you are a fetus, what will happen is that you'll be increasing in length as you grow. What will happen is that this gubernaculum in males will not increase in length. But what will happen is that for females, what will happen is that there's that change. There's that variation. It increases in length as the body length increases. So this is what we are going to see regarding the descent of the testes into the scrotal sac. Now, as the descent goes on, so we have what we call the transabdominal descent. The descent as it moves from where we have the kidney area in the abdomen, as it descends. Okay, and then we have what we call the transinguinal, okay, all the way into the scrotal sac. There are two main hormones which are going to play a role in ensuring that the testis descends into what we call its normal position. So for the transabdominal one, now in the fetal testis, we have two main cells over there, which we are going to see actually when we look at the histology of the testis pretty soon. We have what we call the columnar cells, okay, which are the supporting cells or the testicular cells, otherwise known as Sertoli cell, with the eponym Sertoli cell. And then we also have what we call the interstitial cell of Leydig. Okay, this one is actually epithelial in nature, and this one is actually connective tissue. Okay, so from the connective tissue interstitium, the cells over there known as Leydig cells, okay, over here, and then we have Sertoli cells. Now the Sertoli cells will secrete a hormone known as anti-Mullerian hormone, or Mullerian inhibiting substance or factor. Now this hormone, what will happen is that it will antagonize the generation or the development of what we call Mullerian duct. Okay, the name is anti-Mullerian duct, so it will antagonize the development of Mullerian duct so that it regresses. Okay, because we've learned that this Mullerian duct will be important in the development of the proximal or the upper 2.5 of the vagina together with the uterus, yes, of course, as well as the uterine tube. So once you have those things forming, then the person becomes female. So with the help of Sertoli cells secreting anti-Mullerian hormone or Mullerian inhibiting substance, then it regresses or causes degeneration of the Mullerian duct so that we form a male actually. Now for Leydig cell, Leydig cell will actually produce what we call testosterone. Leydig cell will be important in production of testosterone. Now what this testosterone does is that it will stimulate what we call genitofemoral nerve to produce something we call neuropeptide calcium gene related peptide. Okay, it's actually neuropeptide, which we call it calcium gene related peptide. Now, this peptide will be important in ensuring what we call the transinguinal or the scrotal migration. Transinguinal or scrotal migration. Again, this same testosterone will be important in the development of what we call the Wolffian duct. So that the duct system in males, which we are going to see, yes, talking about the intratesticular duct and of course the extratesticular duct, they will actually develop from what we call the Wolffian duct. So that under the influence of testosterone, then it tells us that the developing fetus is going to be a male. Then apart from the hormone that I've said, calcium gene related, peptide, it is, actually, yes, apart from that one, what will happen is that for the genital swelling, okay, formation, which will be important in generating what we call the scrotum, okay, will be actually coming from the testosterone, will be influenced by the testosterone. Of course, genital tubercle will help in what we call formation of the penis, which we are going to see later. Now, this same, Leydig cells will be important in producing what we call insulin, like, protein 3, okay, which is actually going to be produced under the influence of the gene insulin, like, gene 3. So that what will happen is that it will help in what we call gubernaculum formation, so that this gubernaculum will attach, the testis at one end and pull it into the scrotal sac. So at the end of the day, all these hormones, yes, are going to help in that regard. Insulin, like, protein 3, yes, testosterone, as I've indicated, yes, anti-millennial hormone, together with calcium gene related peptide, helping in both, the transabdominal and then transinguinal, and of course into the scrotal area. Now, one thing that we have to know is that, yes, normally, yes, prior to birth, we expect that the scrotum, okay, should be the dwelling place of the two testes or the testicles. But in some people, yes, what will happen is that the scrotum, as it descends, okay, into the scrotal area, perhaps due to abnormalities in any of these cells producing appropriate hormones, either transabdominal, anti-millennial hormone, or testosterone, yes, then there could be a problem with the descent. So that if you look at the newly born individual, what happens is that there's no testis inside the scrotum or the scrotal sac, so that the testis is hidden somewhere here. So that when we have such a phenomenon, then we call that testis hidden testis or concealed testis or undescended testis, which we also call cryptorchidism. Crypt has to do with hidden. So cryptorchidism, yes, is seen. Yes, it could be seen, in the abdominal area. Yes, even in the inguinal canal, as it goes through the inguinal canal, which we are going to see. Then it can get stuck somewhere, perhaps due to hormone deficiency. Then it can be in the suprascrotal region, so on top of the scrotum, it was unable to descend into the scrotum. Okay, so these are the areas that we can find actually. Then what will happen is that, yes, there could be another situation which we call it ectopic. The word ectopic means finding itself in an area where it's not supposed to be. Now, for cryptorchid testis, what will happen is that, yes, during its normal descent, it was unable to reach its normal position. Okay, so that's why we are finding it in such a place. But with ectopic testis, as it descended, what will happen is that, yes, it was unable to go through its normal channel where it was supposed to actually go through. It was unable to go through the standard routes, okay, going through the inguinal canal all the way into the testis, but rather find itself in some areas where we can have some of these things. For instance, if you look at the lower abdomen over here, okay, this is actually the suprapubic region, okay, on top of the pubis. So we find itself here, and there was no way it should have gone here, because it was supposed to move through what we call the the inguinal canal, within what we call the spermatic cord, but it was not able to actually go through it. Okay, so it tells us that the spermatic cord is actually within the inguinal canal, so it was unable to go through this this inguinal canal into the scrotum. So it finds itself here in the suprapubic area of the lower abdomen. Yes, it can also find itself, in the anterior aspect of the thigh, the femoral region. It can even happen in the perineum, okay, in the perineal area. Yes, all these areas. It can even find itself in the penis. Yes, on the dorsal aspect of the penis, it can be there, or even in the skin around, the scrotal area. So when you have such a phenomenon, then we call it, ectopic testis. Now, some individuals, yes, they are able to even the testis find itself in the scrotal region. They're able to, pull it, and then eventually. So I'm going to show you the muscle which is going to be relevant in that. Mainly being what we call the cremaster muscle. Yeah, they're able to contract it so that they are really able to conceal their testes. We have some children who are able to play with that. Okay, that is what they are able to do. All right, so these are some abnormalities which we can find regarding the descent of the testes, and of course ectopic testes as well. Now, so let's look at this section, a cut through the spermatic cord, okay, so that we reveal certain structures. Now I told you that this is the scrotal sac, okay, the skin over here, scrotal sac, which within it we are going to find the testes, final destination of the testes. Now this scrotal sac, yes, within it, the superficial fascia attaching to the skin actually will be this muscle which we call it dartos muscle. Now it is this dartos muscle that upon contraction, what will happen is that it causes wrinkling around the scrotal sac. Usually during cool conditions, the dartos muscle is actually smooth muscle. It contracts involuntarily, yes, and of course shrinks it causes shrinkage around the skin of the scrotal sac. Now, one other thing that, let me explain this before I even move on. Now, one thing that you have to understand is that why do we need the testes in the scrotum? It should come outside the abdomen. It should come outside the pelvic region. It should hang outside the perineum. So the reason is that, yes, because spermatogenesis, which I'm going to show you, yes, is going to take place optimally under the temperature of about 2 to 3 degrees Celsius, below the core body temperature. The normal body temperature should be around 37 degrees Celsius. So if there's 2 or 3, then it's supposed to take place around 34 up to 35 degrees Celsius. So it hangs outside that to help maintain that temperature. But again, what will happen is that, yes, if let's say under cold conditions, if the temperature becomes too low, then what will happen is that the testes will have to be brought a bit closer to the body so that it receives some warmth, so that it doesn't go way below the 3 degrees Celsius, okay, difference. So that is one thing that is going to take place. However, if the body temperature is too warm, then what will happen is it has to move further away from the body, so that the wrinkling, yes, will be lost through relaxation of the dartos muscle. One other thing too is that, yes, during cold conditions, the testes will have to be pulled quite closer to the body, and it's through the contraction of what we call cremasteric muscle, or the cremaster muscle. Okay, so that upon contraction, this cremaster muscle is actually made of skeletal muscle. So it will contract to pull the scrotum together with the testes closer to the body to receive some warmth. And then the opposite will happen. Relaxation of the cremasteric muscle will happen so that it brings the testes together with the scrotal sac further away from the body, so that it receives less warmth. Okay, so that is that for you with the scrotum. Now, of course, the scrotum will develop from what we call scrotolabial swellings. Actually, that's what we are, or fools. That's what we are going to see. Now, one thing that I want you to know is that apart from this, one other thing too, which is going to help regulate the temperature of the testes. Now we are going to see what we call the seminiferous tubules, which will be the factory for sperm production. as I said, the temperature should be at least 2 it should be 2 or 3 degrees Celsius below the body temperature. Now remember that we have the gonadal artery, which supplies blood to this gonad. The gonad is the testis. Now this gonadal artery is therefore given the specific name testicular artery. Testicular artery. Now look at this testicular artery is actually surrounded by, there's a testicular artery, the reddish one that you find over there, supplying blood to the testis. Now remember that within the blood, we are warm-blooded animals. As well, as a result, the blood which flows to various structures will actually contain the body temperature, or will actually have the core body temperature. Therefore, surrounding these, what we call testicular arteries are some veins which are going to drain the testicular vein, and those veins are known as pampiniform venous plexus, or pampiniform veins, or plexus of veins, surrounding it in this regard. So that what will happen is that as this blood gets to the testis, what will happen is that through countercurrent heat exchange mechanism, that heat within the testicular artery will be exchanged, will move into what we call the pampiniform venous plexus. Of course, you know that because they are surrounding, heat will flow from a region where we have greater temperature to the region where we have lower temperature, to what we call the pampiniform venous plexus, so that it sends it upwards, away from the testis. So that is one thing that you have to know, that the testis, yes, will have to get a temperature below the core body temperature, about 2 to 3 degrees or so below the core body temperature. And this one is achieved, yes, one, because the testis is within the scrotal sac, outside the body. Yes, again, there are some muscles, the dartos muscle together with the cremasteric muscle, dartos muscle being smooth muscle, cremaster muscle being skeletal muscle, helping to actually bring the the testis closer to the body or away from the body when the need arises. And then again, the nature how the testicular artery is surrounded by the pampiniform venous plexus to help in that countercurrent heat exchange system. So that is what we are going to see. Now we can see that yes, there's a testis. The testis will have a small outportion structure known as appendix of the testis. Then we also have surrounded posterolaterally to the testis, we are going to find what we call the epididymis. We are going to see all these structures pretty soon, lying over here. That's the epididymis. Plural is epididymis. Now the testis is a solid organ. Yes, it actually lies in a serous membrane, and this serous membrane is known as tunica vaginalis. Tunica vaginalis. Now this tunica vaginalis, the one which lies on the wall within which the testis lies, is known as parietal layer of the tunica vaginalis, and the one which is intimately associated with the substance of the testis is known as the visceral layer of tunica vaginalis. Of course, visceral has to do with organ. Now one other thing too, when I go deep to this testis, when I remove the tunica vaginalis, I'm going to see what we call tunica abugenia, then tunica vasculosa. So we are going to see all these ones as I move on. Alright, now so let's look at blood supply to the scrotum. We've seen the blood supply to the testis being testicular artery drained by the pampiniform venous plexus. Now for scrotal sac, yes, it will be drained by two arteries, one being the anterior scrotal artery. This anterior scrotal artery will actually drain into what we call external pudendal artery, which will also drain into what we call the femoral artery, which is actually continuation of the external iliac artery. Then we also have what we call posterior scrotal artery, which will actually drain all the way into what we call internal pudendal artery. So while anterior scrotal artery draining into external pudendal artery, that of the posterior is draining into the internal pudendal artery. And of course, they will be drained by the corresponding vein. So of course, by anterior scrotal vein, and of course anterior posterior scrotal vein. Now regarding lymphatic drainage, what will happen is that yes, the testis together with the scrotum, they will actually drain into what we call superficial inguinal lymph nodes. Superficial inguinal lymph nodes. Okay, that is how the testes together with the scrotum will drain. Okay, so they actually drain in an ipsilateral manner, so that if it is what's the name, if it is the testis, then the testis is actually going to drain actually into the para-aortic nose, just like the ovary into the para-aortic nose. Okay, then of course the scrotum will actually drain into what we call the superficial inguinal lymph nodes. So these are one other thing, things that you have to actually learn about these ones. But what will happen is that sometimes the scrotum, instead of draining into the superficial inguinal lymph node, from superficial it will go to the deep inguinal lymph nodes. Okay, but what will happen is that for sometimes it can drain directly into what we call the deep inguinal lymph nodes. So importantly, what will happen is that for the gonads, they are draining to the para-aortic nose. For the scrotal sac, they are actually draining into the superficial inguinal lymph nodes, which will drain further into the deep inguinal lymph nodes. But sometimes can drain directly into what we call deep inguinal lymph nodes actually. All right, now regarding lymphatic drainage, one other thing that you have to understand over here is what we call hydrocele. So what will happen is that lymphatic fluid can actually accumulate around the testis inside the scrotal sac, and that is what we call hydrocele. Okay, so it's actually a form of lymphedema actually, so lymphatic problem. For instance, if you are suffering from elephantiasis, yes, you can have such a phenomenon, draining into the scrotal sac around over here. Now this one is actually not retractable, unlike you find in the case of hernia. Now for hernia, it's actually a content of the abdomen which is actually moving into what we call the scrotum, either directly or indirectly. So when you look at hernias, you actually understand that one. But this one is just fluid, okay? Lymphatic fluid actually seeping into this place. All right, now one thing too that I want to say is that now when you look at the testis could actually be inflamed, and that's what we call orchitis. Now sometimes the inflammation could be bacterial infection causing this inflammation, who affects this structure which lies on the posterolateral aspect of the testis, which we call the epididymis, together with the testis itself, and therefore we call that one epididymo orchitis. So if you hear of epididymo orchitis, then it's inflammation of the testis together with what we call the epididymis. Now there is this disorder which sometimes it happens to children around less than 3 years. It happens to them, but sometimes it gets resolved on its own. But usually it's most of the time it has to be corrected through surgery, and that is what we call testicular torsion. Now in testicular torsion, then what will happen is that the testis rotates. Okay, there's rotation of the testis, and this rotation will cause twisting of the spermatic cord. Okay, cause twisting of the spermatic cord. And the spermatic cord will actually contain the structure being the artery, the structure over here being what we call the gonadal artery, being the testicular artery actually. So when there's that twisting, then what will happen is that the lumen is occluded somewhat, so that blood supply to the testis is impaired. When such a thing happens, then it means that the testis is not receiving enough nourishment. It may begin to die. The cells over there may begin to die, and it results in severe pain, and swelling, around this area. And as I indicated, usually surgery will have to be in place to correct this torsion. So if you are someone who loves that your testis is played with by a lady, then what will happen is you have to be very careful so that you don't cause torsion over here. Otherwise you will suffer a great deal of pain, and it is not going to be helpful at all. Now if we take a sagittal section, okay, through the substance of the testis, then we are going to see this. Now I'll actually go through the intratesticular and extratesticular duct system, But for now, I told you that yes, we are going to have what we call, when you look at the testes, which will hang in the scrotal sac, we are going to have a visceral layer as well as parietal layer of tunica vaginalis, and deep to the tunica vaginalis will be a layer of dense irregular connective tissue known as tunica albuginea. Tunica albuginea. Remember that we've seen tunica albuginea when we looked at the ovary, which was deep to the simple cuboidal epithelium, which was actually the mesothelium of the ovary, which we used to call it, the germinal epithelium. This tunica albuginea is, going to be deep to the serous membrane, being tunica vaginalis. So look at this. Now, this tunica albuginea begins to, invaginate the substance of the testes. Okay, so that these invaginations are the septa, which we find in the testes. Okay, so that between two septa, we are going to have lobule of the testes. So the tunica albuginea will invaginate the substance of the testes, dividing the testes into lobules. Now, each lobule will contain 1 to 3 these convoluted structures, which we call them seminiferous tubules. Okay, so these are the convoluted structures that we find. So each lobule will have 1 to 4 seminiferous, tubules, which we are going to see. So that is one thing that you have to know. Now, this is the gateway of the testes. Okay, so this area is known as mediastinum of the testes. Mediastinum testis. So we are actually going to look at all these things, okay, as we move on. Now, let me even just try to show you this. Now, this over here, which I said is the epididymis. The epididymis is actually divided into a head portion. We have the body or the corpus part, and then you have the tail. Now, we know that the epididymis is going to store sperm. Okay, apart from that, yes, it's going to concentrate sperm, and of course, you're going to release them when the need arises. Now, if you are having head, body, and tail, then it means that the most functional sperms will be in the tail region. So as we move on, we're actually going to see all these things. Now, so this is actually, yes, a section through the testes. As I showed you, we are going to have what we call, the tunica. Yes, tunica, we are going to have a layer of serous membrane, being the tunica vaginalis. Then we have what we call tunica albuginea. Okay, so if we take the substance of the test, we are going to see tunica albuginea. Below that tunica albuginea, we are going to have a layer of loose connective tissue rich in a lot of blood vessels, which we call a tunica vasculosa. Now, these ones represent the factory. These ducts, the convoluted ducts that you find over here, represent the factory for sperm production, and that is what we call seminiferous tubules. Tubules. Now, this seminiferous tubule, yes, will have what we call an epithelial lining. That epithelial lining is described as complex stratified epithelium. Okay, we'll have a complex stratified epithelium. Now, in the epithelial lining, in the complex stratified epithelium, we have what we call the stratified portion and the non-stratified part. The stratified part will contain what we call the cells of spermatogenic series, okay, which of course will eventually give rise to the functional spermatozoon. Okay, so that is what we are going to see over there. But what happens is that we have the non-stratified part being what we call Sertoli cell, or the supporting cell, or the sustentacular cell, which we are going to actually see all these ones. Now, I told you that yes, the septi, which we are going to see from invagination of the tunica albuginea into the substance of the testes, will divide the testes into actually about roughly 250 lobules. Okay, so bear that one in mind. Now, so as we move on, so let me just digress a bit and then go through what we call spermatogenesis, which is sperm production. Now, unlike in females who are going to have fixed number of oocytes, okay, which we call them, the oogonia, which will go through various development processes, which we already looked at that one. For males are able to produce sperm continuously throughout their lifetime, okay, and they're able to do that because their stem cell always goes through what we call mitosis, giving rise to the stem cell. Okay, and of course may also give rise to those one which will proceed in the spermatogenic series. So actually, let's look at this now. The sperm cell of the stem cell of the spermatogenic series is actually known as what we call type A duct spermatogonium. Type A duct spermatogonium. Now, this type A duct spermatogonium will undergo mitosis to give rise to a lot of type A duct spermatogonium. So it just goes through mitosis, giving rise to a lot of type A duct spermatogonium. It can go through several mitoses, and all those mitotic divisions will give rise to what? Type A duct spermatogonium. Now, what will happen is that, yes, this type A duct may also undergo mitosis to give rise to what we call type A pale spermatogonium. Okay, so what happens is that we can give, depending on the state of the body needs. If the body needs a lot of spermatozoan, okay, because we are ejaculating all the time, then what will happen is that from type A duct, they will just go through mitotic division to give rise to what? Type A pale spermatogonium. Okay, that's what we are going to see. Otherwise, if you don't need a lot of sperm, if you are someone who don't have sex often and you don't need it more, then what will happen is that when type A duct spermatogonium undergoes mitosis, it will just be giving rise to a lot of type A duct spermatogonium itself, with few type A pale spermatogonium. So assume we want to go through the normal trend, then what will happen is that type A duct spermatogonium will undergo mitotic division to give rise to what we call type A pale spermatogonium. Type A pale spermatogonium. And type A pale spermatogonium also go through mitotic division to give rise to what we call type B spermatogonium. Type B spermatogonium. Then type B spermatogonium will undergo mitotic division to give rise to what we call primary spermatocyte. Now remember that all these divisions are happening at puberty. They are not happening, in utero. It's at puberty, somewhere in 15 or so. That's where all these divisions, mitotic divisions are going to take place. Now, when we get to primary spermatocyte will now go to the first meiotic division. So, primary spermatocyte, so it means that type A duct, type A pale, type B spermatogonium are all diploid. They will have 2N, all diploid. Okay, so if I diploid cell, then it tells you that the cell, the body's immune system, recognizes you. Okay, won't mount any immune response to you because diploid cells were introduced to the immune system during early development, okay, of the immune system. But what will happen is that I told you that primary spermatocyte will be actually the intermediate. Okay, will serve as a boundary between what we call I'm going to show you some now, just to digress and show you this. This is the lumen of the seminiferous tubule. These represent the epithelial lining of the seminiferous tubule, which we call it the stratified, complex germinal epithelium. Complex stratified epithelium. It's actually germinal epithelium. This one is actually a very good name for it because they are going to give rise to the functional spermatozoa, functional gametes of the male. Okay, so that is what we are going to see. Now, this is the lumen, and as we move this, we are moving away from the lumen. So as I move closer to the lumen, then we have what we call adluminal compartment. Adluminal compartment. Adluminal compartment. Because I'm getting closer to the lumen. Now, further away over here will be what we call abluminal, or the abluminal. I'm moving away. Abluminal, or the basal compartment, Now, the basal compartment over there, you will find only diploid cells. So you may be seeing the type A duct, type A pale, type B spermatogonium will be here. Okay, and there wouldn't be any immune response mounted on them. Why? Because they are diploid cells and the body recognizes them. But from primary spermatocyte, where the first meiotic, so primary spermatocyte goes through the first meiotic division, okay, and therefore will serve as boundary between what we call the adluminal compartment and the abluminal compartment, or the basal compartment. Okay, so we have what we call the primary spermatocyte going through the first meiotic division, to give rise to what we call the secondary spermatocyte. Secondary spermatocyte. So the primary spermatocyte is actually going to be haploid, and then secondary spermatocyte is also going to be haploid. It will also go through what we call the second meiotic division. Remember that first meiotic division is actually a reductional division, so from diploid state to haploid state, okay, to become haploid. And then secondary spermatocyte is also haploid. So secondary spermatocyte goes through the second meiotic division to give rise to some cells. We call them spermatid, which we are going to look at their structure. They are actually oval shaped generally, but as they grow, then they begin to elongate, shedding most of their cytoplasm as well. So that is, that for you. So over here we are going to find the diploid cells, okay, which represent the spermatogonium, either type A, duct, or whatever. Then we have the primary spermatocyte, okay. Then of course, as we move on, now we are saying that there should be blood, okay, or the interstitial fluid, okay, shouldn't reach what we call the abluminal compartment, okay. And the reason is that we are able to maintain that, yes, because there are these non-stratified cells which are columnar shaped, and these cells are known as the Sertoli cells or the sustentacular cells. Now they form what we call, they give off process. So this is sustentacular cell will give off a process, this one will give off a process to form what we call a tight junction, so that fluid coming from the interstitium, okay, won't reach the abluminal compartment. And because if there is leakage to this tight junction which will be created, then what will happen is that it may lead to infertility in males. Okay, so that is one thing that we are going to see. Now this is actually spermatogenesis, but this spermatogenesis will actually be completed, okay. I told you that this one, secondary spermatocyte, will undergo second meiotic division to give rise to what we call spermatid. Spermatid will therefore go through what we call cytodifferentiation process, okay, shedding off most of its cytoplasm, becoming elongated, and then becoming what we call the mature spermatozoon. Okay, so that is that for you. These Sertoli cells, okay, let me just explain what they are going to do again. Apart from forming what we call blood testis barrier, yes, they will also be important in nourishing cells of the spermatogenic series. They nourish them. So they are the nursing cells. They would nurse the cells of the spermatogenic series. Apart from that, they will also secrete what we call androgen binding protein into the lumen. Why do we need that? They will secrete androgen binding protein into the lumen over here, so that there will be another cell which I'm going to tell you, which are known as the interstitial cells of Leydig, the Leydig cells. They will, under the influence of interstitial cell stimulating hormone, will produce what we call testosterone. Now this testosterone will accumulate in the lumen of the seminiferous tubule to cause spermiation, okay, to move these spermatozoon, okay, or spermatus into what we call the lumen, okay, into the lumen, so that they can actually move all the way into the various duct systems which we are going to see, actually to the straight tubule or tubule recti, eventually into recti testis and what have you. I'm going to go through that one. So that is one thing that is going to do. Yes, apart from that, if there is any defective cell over here, because it's nursing them, then they could be phagocytosed, okay, by the Sertoli cells. Then they also produce what we call inhibin, okay, which will inhibit what we call follicle stimulating hormone secretion. Now remember that all these follicular growths, all these growths by the cells of spermatogenic series will be under the influence of follicle stimulating hormone coming from the anterior pituitary gland. So if there are enough cells which are being produced by the epithelium, then what will happen is that the Sertoli cells will secrete inhibin to cause the slowing rate of the secretion of follicle stimulating hormone by secreting actually what we call inhibin. Then it may also secrete some paracrine substances, okay, to actually affect these interstitial cells of Leydig, okay, so that to influence their secretions as well. So mainly that is one thing it's going to do. But I told you prior to, when we began this tutorial, I told that it's also going to produce, these Sertoli cells will produce what we call anti-Mullerian factor or Mullerian inhibiting hormone, which was going to ensure that the male, the fetus actually grows to become a male. Okay, we've seen that one too. So let's move on, and as we move on, we'll look at other things. So here we are. This is actually a histological section trying to show you the lumen of the seminiferous. This just a magnification of a lumen of seminiferous tubule over here. So here you have, some of the spermatozoa being here. Then as you get more closer to the lumen, then the more mature ones, the more haploid ones will be getting closer to the lumen. And those ones which are more diploid will be getting, away from the lumen to the, abluminal compound, the basal compartment. Of course, in between these cells, and I told you in between these cells, there will be a, connective tissue, and that's the interstitium, connective tissue, interstitium. By separating the connective tissue will be a basal lamina, basement membrane will separate them. But you see, this is one of the cells that you find over here, being the Leydig cell, interstitial cell of Leydig. And I told you that the Leydig cells will secrete what we call testosterone. Now remember that testosterone will be very important in increasing the libido of both male and females. Yes, remember that. When you look at the reticular layer of what we call, when you look at the endocrine system of, actually the cortex of, the suprarenal gland. Yes, we are going to have what we call, the reticular layer will produce what we call adrenal androgens. So generally, testosterone will be important in actually increasing libido in both males and females. Apart from that, the male sexual characteristics, okay, will be coming from testosterone. Remember that all the organs of the male reproductive system, for them to grow, okay, the testes, the testicular ducts, whatever, for increase in size of even the penis, will be under the influence of testosterone. Now remember that the interstitial cells of Leydig, or the Leydig cells, were actually going to perform their function under the influence of what we call interstitial cell stimulating hormone, also coming from anterior pituitary gland. Now this interstitial cell stimulating hormone we saw in females was called luteinizing hormone. So if you hear luteinizing hormone, we are still looking at the interstitial cell stimulating hormone. Now, one thing that I have to tell you over here, which of course I've already told you, is that Wolffian duct, yes, will give rise to all the male testicular systems, which, the duct system, which we are going to see actually. Okay, so that is that for you. Now, I told you that, yes, so this is actually early spermatid, which is roughly, oval in shape. Okay, so it will go through what we call spermatogenesis, that's cytodifferentiation. But they are released, okay, into, they are released, which will be aided by what we call Sertoli cells, into what we call the lumen of the seminiferous tubule, will be called spermiation. So remember that there's difference between spermiogenesis, spermatogenesis, and spermiation. Get to understand that. So this spermatid, yes, will have a roughly oval shape, yes, with the centrally, so that it can actually help in formation of the flagellum, okay, through, microtubules also helping in that regard. Yes, so eventually there will be shedding of most of the cytoplasm of the spermatid. The nucleus will also, shrink, okay, will become a bit smaller, okay, and then, at the end of the day, for a spermatid, it will have what we call vesicular, nucleus. So that the vesicular, nucleus, say that, the nucleus centrally becomes a bit pallor, and then the outer rim is a bit, darker. So excess cytoplasm, as we said, will be shed off, and then eventually we are going to get, the functional sperm. So the sperm will actually have the head, the middle piece, and of course the tail. Now, so actually when we look at the mature sperm, as we've seen, the mature sperm is known as spermatozoon. Okay, spermatozoon. Now this spermatozoon, yes, will actually have, yes, head piece. Okay, so within the head piece we'll anteriorly we'll have what we call the acrosomal cap. Okay, we'll have the acrosomal cap. Now within the acrosomal cap, we will have a lot of hydrolytic enzymes. We will have a lot of hydrolytic enzymes, and these hydrolytic enzymes, yes, will help. For instance, will contain raminidase, hyaluronidase, so that they're able to actually secrete those enzymes to break through what we call the barrier over here. I'm going to tell you the barrier of the oocyte, so that it can actually penetrate the oocyte. Now, what will happen is that even in the nucleus, again, there's nucleus as well in the headpiece, which will contain the DNA material. This cell will be haploid, so it will have 23 chromosomes. Actually, it's haploid. That's what we are going to find here. And yes, you have what we call the middle piece. Now, the middle piece will be endowed with a lot of mitochondria. Okay, so that, yes, utilizing what we call fructose, it can metabolize the fructose to generate energy. Remember that the sperm cell will not rely on glucose to generate energy, but will rely on fructose to generate energy. So that is what we are going to find with what we call the middle piece. Mainly, that's what we are going to find over there. Then, of course, we are also going to have the tail piece. Now we have the principal tail piece, and of course, the end piece, or the terminal, piece of the tail, so that, yes, it is flagellum. Yes, it's animal-like actually, so that it can actually help propel, the spermatozoon, okay, as it goes through the seminal fluid, even the vaginal fluid, to fertilize the oocyte. So that is that for the spermatozoon, functional sperm, that you have to know. But one thing that I have to tell you is that the barrier, okay, which the sperm will have to break through before fertilizing the oocyte, so that the nucleus over here, they will combine, forming what we call the pronucleus, the male pronucleus and the female pronucleus, eventually for fertilizing to occur. It has to penetrate these remnants of the follicular cells, which was surrounding the oocytes, attaching to the zona pellucida, being the corona radiata. It has to penetrate it. It has to also penetrate through what we call the zona pellucida, with this translucent belt over here. It has to penetrate it. Then it has to also penetrate what we call the membrane, the cell membrane or the plasma membrane of the oocyte, before it can get to the cytoplasm, and eventually there are nuclei fusing to form the male and female pronucleus for fertilization to occur. So that's one thing that you have to know. Now, by so doing, what will happen is that, yes, this spermatozoon is actually not going to fertilize this oocyte, secondary oocyte, except it goes through a process we call capacitation. So that I have some glycogen material surrounding it. It has to be removed. The acrosomal cap will have to be well positioned in that regard. Okay, and it's actually going to take place within the female reproductive system, within the vagina. Actually, it's going to take place there, and that's capacitation. And that's why in vitro studies, what will happen is that, yes, the spermatozoon, the functional spermatozoon, is unable to fertilize the secondary oocytes unless it's treated with some ionic salt solutions, so that the glycoprotein material surrounding it will be removed, so that it can actually be able to fertilize this secondary oocyte. Okay, so that is that for you. All right, now one other thing too that we have to know, yes, is that once the seminiferous tubules, which we've seen over here, seminiferous tubule being actually about 30 to 70 long, yes, just one of them being about 70 to 30 to 70 centimeters long, usually having a diameter of roughly 200 micrometers, 200 micrometers. Okay, so if you combine all of them, then it tells you it's very long. It's a very long structure, this seminiferous tubule, but it's actually convoluted so that it can be packed inside the testis. Now, once the seminiferous tubule has produced this spermatozoon, then what happens is that inside the lumen of the seminiferous tubules, they will be moved into this tubule. So you can see these tubules are not convoluted, and therefore we call them straight tubules or tubuli recti. Tubuli recti. Recti has to do with straight. Tubuli recti. And this tubuli recti, yes, will also eventually empty into this anastomosis channels or connections over here, which is located in the mediastinum testis, with the gateway of the testis being known as reti testis. And then from the reti testis, these ducts, yes, they also, open into about 10 to 15 of these ducts, known as efferent ductuli, because they are leaving the substance of the testis, so we call them efferent ductules or ductuli efferentes. Efferent ductules. And these efferent ductules, part of it will be inside the testis, and then part of it will be outside the testis. So what I'm saying is that for intratesticular ducts, apart from the seminiferous tubule being the factory for sperm production, we are going to have the straight tubule or tubuli recti. We are going to have reti testis and some most proximal portion of what we call the efferent ductules. Now the remaining part are outside the testis, so we call them extratesticular ducts. So beginning extratesticular ducts, the most distal portion, of the efferent ductules or ductuli efferentes. Then we are going to have what we call they will actually open into a single duct. So you can see this one, they are the efferent ductules. This one I can count about 5, but actually 10 up to about 20. You can have those variations in there. Now they will open into a single duct, so you can see this is actually a single duct. This single duct is also highly convoluted. This single duct is known as ductus epididymis. So this region over here is the epididymis, but the epididymis is having a single duct, which the efferent ductules open into. Okay, and then from there we are going to have what you call ductus deferens. But this ductus deferens, sometimes they call it vas deferens. Now vas deferens is actually a misnomer. The reason is that usually when we say vas, it has to do with vessel. So originally when they identified it, they thought it was a blood vessel, so they gave it the name vas deferens. But it's actually a duct for conveying the seminal product, and therefore we call it ductus deferens or the sperm duct actually. Then we also have what we call we'll have what we call the ampulla of the ductus deferens, joining what we call the seminal vesicle to become the ejaculatory duct, and then draining into what we call the prostatic part of the urethra, and then eventually to the external environment or into the vagina if copulation is going on. So we are going to actually go through this duct system. So as I told you, remember that I told you that the epithelial lining of the seminiferous tubule was complex stratified, having what we call stratified part and a non-stratified part. The stratified part actually represented that portion which we call it the cells of the spermatogenic series, giving rise to the sperms of the spermatogenic series. But what will happen is that we also have the non-spermatogenic series, which is non-stratified, and that is the one we call the Sertoli cells, the columnar shaped ones. Therefore, the straight tubules, which just continues from the seminiferous tubule, is actually going to have only the Sertoli cells. So the straight tubule will have simple columnar epithelial tissue lining it. Okay, so that is what we are going to see with the straight tubule. And then the straight tubule, you see, you have this kind of lumen, having all those things. So you may have some, some what do you call spermatozoa, actually in there. So how do you differentiate it? All that you are going to see is that you have connective tissue interstitium. All right, you have what we call it. You have Sertoli cells, yes, but you don't have cells of the spermatogenic series. Cells of the spermatogenic series will be absent. So that is something that you have to know regarding the straight tubule or tubuli recti. Now, just to show you again, we are going to have the seminiferous tubule. There's a testis seminiferous tubule. Yes, we are having the straight tubule, different ductiles. It's not going to be stratified as we saw in the case of the seminiferous tubule. There's going to be a single layer, non-stratified layer, simple squamous epithelium. Okay, is what we are going to find. All right, so this area is actually trying to show you the mediastinum testis, the mediastinum testis. Now, within the mediastinum testis, which is the gateway to the testis, we are going to find these areas, channels, which I showed you, several channels. This area is highly vascularized, so you can see this reddish area that you find over here, highly vascularized, although you can't see it over here, but the epithelial lining is simple cuboidal epithelial tissue, simple cuboidal epithelial tissue. Now, this simple cuboidal epithelial tissue is bearing a single cilium, also endowed with what we call microvilli, so that at the end of the day, it can help propel, okay, the spermatozoa as they go through, they go out of the testis. Okay, then there will be testicular fluid as well. So absorption of testicular fluid, yes, will be done over here as well, by the rete testis. Okay, we'll begin absorption of the testicular secretions. All right, now this represents what we call ductally efferentis. So I told you that from the rete testis we go to ductally efferentis or efferent ductilis, which I told you about 10 to 20 ducts of these ones. Now, one thing is that yes, this one is going to have what we call simple columnar epithelial tissue, okay, which are going to have two different cell types. Some of the cell types will bear cilia, others will not bear cilia. Okay, so some of them are ciliated, others are non-ciliated. That is why you see that the lumen appears to be rough, somewhat. Now, another thing too, that for you to identify this is that it's going to have almost stellate shape appearance. Okay, it will have stellate shape appearance, or something you call scalloped lumen. It will have a scalloped lumen. Its lumen is strewn into these folds, appearing like you have some semicircles. Okay, so it's having this scalloped lumen actually. So that is one thing that you have to know regarding the ductally efferentis. Then the next one, now remember that these ductally efferentis are going to drain into a single duct. Okay, about 10 to 15 of these ductally efferentis are going to drain into a single duct known as ductus epididymis. This duct is actually about 30 to 45 centimeters long. And I told you that this ductus epididymis, this epididymis, will actually lie on the posterolateral aspect of the testis, and of course it's going to receive duct coming from the efferent ductilis that we have seen. Then it's going to have a head, body, and tail, as I showed you, with most of the most functional spermatozoa being in the the tail, so that if you want to do in vitro fertilization, for you to get more mature sperm, then you have most mature sperm, you have to go into the tail part of it. Yes, then of course we are also going to be endowed with, now the epithelial lining will be pseudostratified columnar. So you can see that the cells over here, not all of them are reaching the base. So pseudostratified columnar epithelium bearing these stereocilia. Now if you look at the lumen, there's just magnified portion of just one of the profiles. Now, one thing is that when you take the profile, when you cut through the ductus epididymis, because of its highly convoluted nature, you may be seeing several profiles of the ducts. Okay, it doesn't mean that it's having a lot of ducts, it's just a single duct, but because of the highly convoluted nature, you are going to see several of these profiles. Now, this is just one of the profiles that you see. These represent actually the spermatozoa in its lumen, Okay, now the sericilia is going to actually perform absorptive function, trying to absorb most of the testicular fluid from the spermatozoa, so that it helps concentrate it, because after all it's going to help concentrate it and it's going to store it as well. Now, one thing too is that these are the principal cells, the ones that you see more closer to the lumen. Now you can see these ones which are almost cuboidal, but they're also supposed to be columnar. They represent what we call the basal cells. So there are two cell types over there which you may see. You may see the principal cells, and then you may see also the basal cells as well. Then this is where actually the muscular parts of the male reproductive system actually begins. Yes, this is also going to continue from what we call the ductus epididymis, and therefore this one also have pseudostratified columnar epithelium having stereocilia. Now look at it, it's also thrown into folds so that we get this kind of star-shaped lumen. Only that this one, yes, is going to have three tunics. We can actually identify the three tunics: a mucosa, muscularis, and of course adventitia. So the mucosa is going to consist of the pseudostratified columnar epithelial tissue with stereocilia, together with the underlying lamina propria, because we know beneath every epithelial tissue we are going to have loose connective tissue of lamina propria. Then beyond that, we are going to have this muscle coat. Now this muscle coat is actually made up of three layers: an inner longitudinal, middle circular, and outer longitudinal. So if you look at it closely, because there's a transverse section through the ductus deferens, or the sperm duct, or the vas deferens, what happens is that the inner longitudinal, because the muscle fibers are running in longitudinal manner, as you cut it through transversely, then you disturb the muscle fibers so that you can see these pieces of muscle fibers, these profiles. But because the circular one is running in the direction of the cut, because the transverse cut, they won't be disturbed. And then of course the longitudinal, outer longitudinal is also going to be disturbed. But what you see is that there's high infiltration of connective tissue within the muscle, which is actually characteristic of both male and female reproductive tracts, okay, which we are going to see. So that is that for you. Now this muscle is actually very thick, okay, thick muscle core, so they can actually propel, okay. This is actually the most muscular portion of the male reproductive system, okay, so that can actually propel the semen through a bullet like fashion during ejaculation, so that I can push it into the urethra, into the ejaculatory duct, and of course into the urethra. And of course its outermost will be by what we call adventitia, okay, so that they can attach it to the surrounding structures, okay, can glue it to surrounding structures. So that is the ductus deferens. So there we are. Here is the urinary bladder, okay. Here we are having the prostate. Of course over there we are going to have the urethra, but what we are going to see is that this is the ductus deferens I was showing you. Now the terminal portion of the ductus deferens actually gives a dilatation, okay, some kind of thickening over here, as you can see over here. This is known as ampulla of the ductus deferens, and this is the seminal vesicle. The seminal vesicle will actually join the ampulla the ampulla of the ductus deferens to become what we call the ejaculatory duct, So we are actually going to go through all these ones. But let me actually explain this. Now if you look at the seminal vesicle, as you can see, they are paired, okay. There's one on the left side, there's one on the right side, and they actually lie on the posterior wall of the bladder. So there's a bladder, they lie on the posterior aspect of the bladder, and the seminal vesicle will actually contribute about 60% of the volume of the semen, okay, in the ejaculate. That's what we are going to see. Now this seminal vesicle will actually contain a lot of fructose. I told the sperm is going to dwell on fructose. That fructose is coming from the seminal vesicle, okay, so that that is what it's going to do. Yes, it will also contain what we call fibrinolysin. It will contain what we call fibrinogen. Fibrinogen in the cascade of the clotting pathway, yes, is very important in blood clotting. So it tells us that immediately after ejaculation, then the ejaculate will have to be clotted transiently, okay, before capacitation. Will have to be liquefied by some secretions coming, for instance fibrinolysin coming from the prostate gland, which we are going to look at it, so that it helps in capacitation, okay, actually. Then the prostate, the seminiferous, the seminal vesicle will actually also contain what we call secretions, will contain what we call prostaglandins, okay, which will actually help decrease the cervical mucus so that the sperm can actually penetrate, okay, the cervix area to get to the the womb and then into the the fallopian tube for fertilization to occur. Okay, so actually these prostaglandins also aid in peristalsis of the uterus so that it can help admit the sperm as they go into the uterus. Okay, so we will actually look at this seminal vesicle. We look at its histology as we move on. But if you look at this prostate gland over here where it's sitting beneath it, this prostate gland, we describe it as being donut shaped, okay, roughly somewhat triangular in shape, as you can see, and it will actually surround the proximal portion of the urethra, but it's actually just below the urinary bladder. Yes, its secretions will actually contain what we call citrates, and citrate will be very important, yes, in ensuring that the pH of the semen is roughly 7.2 to roughly 8.0. Okay, 8 pH should be around that area. Then it will also contain what we call proteolytic enzymes, so that after what we call the coagulation, okay, production of the fibrinogen by the seminal vesicle, prostate will produce what we call proteolytic enzymes, for instance fibrinolysin, so that it can break the clots, okay, so that the spermatozoa are free to actually fertilize. Yes, within the prostatic secretions we also contain what we call zinc. Zinc is very important for sperm vitality, yes, because it's going to make the sperm viable, okay, by inhibiting what we call DNase activity, so that DNase activity will not break down the DNA of the spermatozoon so that it's inactivated. So that yes, the prostate secretion, the seminal secretion, if you are not have a seminal vesicle secretion, if you are not having it, then it's less likely for you to actually produce viable sperms which are likely going to fertilize female. Now within the prostate too, which you get to know, which of course most males around age 40 upwards, they will have to actually get tested for that antigen, and that is what we call prostate specific antigen, so that you check to know your prostate level, whether you are going to suffer from prostatic carcinoma, which you are going to actually look at all these things. Then what I'm saying is that the PSA, okay, in the semen, in low concentrations of semen, remember that PSA normally is supposed to be 0 to about 4 nanogram per ml. That's the maximum value. But if the prostate is actively producing certain things, if you're actually ejaculating all the time, if you are actually riding bicycle all the time, taking in a lot of yogurt, sleeping with different women, then your chances of actually having high content of prostate specific antigen in your blood will be high. But normally in physiological levels, it's going to help actually in the liquefaction of the semen, making the semen more thinner so that the sperm can actually go through it. So that is that for you, but we will actually look at the remaining portions of it. So as I said, the ampulla is a dilated portion of the vas deferens. Ampulla even having more thicker muscle coats, okay, inner longitudinal, middle circular, outer longitudinal, that's the adventitia with blood vessels in there. Then what will happen is that the only difference that you find over here is that its mucosa, yes, you are going to have pseudostratified columnar epithelial tissue with stereocilia, but its mucosa will be thrown into several folds, into primary, secondary, and even tertiary folds over here, so that the lumen is somewhat occluded, so to speak, having this kind of lumen appearance. So once you see this, then you know that you are looking specifically at the ampulla of the ductus deferens. Okay, so this is actually the seminal vesicle. Okay, so if you look at the seminal vesicle, yes, it's also going to have two muscle coats. Actually, it's going to have inner longitudinal, okay, and outer circular coat, inner longitudinal, outer circular muscle coat. But what we are going to see is that its lumen will have these sound of circles making the lumen appearing that it's having this kind of honeycomb appearance. So if you are having this honeycomb appearance, then it's likely going to be the seminal vesicle. Of course, the seminal vesicle will have, pseudostratified columnar epithelial tissue, but sometimes it can be simple columnar epithelial, tissue. So the striking thing is these mucosal folds giving this honeycomb-like appearance. These mucosal folds will give primary, secondary, and even tertiary, so that it gives this, mucosal, honeycomb appearance. Okay, so that is that. So this actually the examination. Now, usually if a doctor is suspecting prostatic hyperplasia, benign prostatic hyperplasia, or let's say prostate carcinoma, then what will happen is that it's one of the examinations. He will go through what we call digital rectal examination, and with a gloved finger or gloved hand, he or she puts the finger into the rectum, so that the posterior aspect of the prostate, is related to the ampulla of the rectum, so that I can feel it, describe the consistency, the volume, everything over here. Okay, so that is one thing that you have to know regarding the prostate gland. Now, over here beneath this prostate gland, okay, actually in the deep perineal pouch, okay, in the deep perineal pouch, actually, around where we have what we call the perineal membrane, okay, so deep urogenital diaphragm actually. So we are going to find what we call the corpus gland, or the bulbourethral gland, because it's around the bulb of the penis and it's around the urethra, bulbourethral gland. So we have that gland too over there. Now that gland is just we say it's pea size, okay, it's shaped like a pea, and what will happen is that it's going to produce this clear fluid. Prior to sexual intercourse, upon stimulation, what will happen is that you realize that there will be some kind of fluid coming out of the penis prior to sexual intercourse during the foreplay. That fluid, that alkaline fluid, which will wash, okay, the urethra from any traces of urine, because the urethra is going to perform both genital, it's going to be a genital urinary structure. Urine will go through it as well as semen. So during ejaculation, prior to ejaculation, what will happen is that this one will have to wash it, okay. And some people say when you have sex and you have a lot of phlegms in you. That's not true. It's actually a normal physiological phenomenon that takes place during arousal, so that you wash the urethra, okay, prior to sexual. It's also going to be important so that it can help in lubrication of the introitus of the vagina prior to intercourse. So that is one thing that we are going to see actually. Now, if I'm interested in the prostate gland, actually, because prostate gland will contribute about roughly close to 30% of the seminal secretion, and we mentioned the components of the prostate gland, the constituents that it is actually going to contain. Now, this prostate gland, yes, as you can see, will have histologically will have three main zones. So these three main zones are the central zone. Now, the central zone will contain about 25% of the functional zones of the prostate, okay. And what will happen is that you can see it is surrounding this. Now, I told that this is the seminal vesicle, okay, and then of course we have the ampulla of the vas deferens or ductus deferens, becoming what we call the ejaculatory duct. Okay, so what happens is that surrounding the ejaculatory duct is the central zone. Okay, now this central zone, the ducts, okay, that open into the prostatic urethra. This is the urethra. They have to empty their product in here through the duct. Now the ducts they empty actually obliquely, in an oblique manner. Now this oblique manner makes it very important, so that this central zone is actually very resistant to carcinomas or to reflux of urine into this area. Okay, so that is one thing that you have to know. Then, of course, the next one will be what we call the transitional zone. Okay, this blue area is the transitional zone. Now this transitional zone, yes, will actually occupy about 65% of the total volume of the functional zone of the prostate. And this one, it's more or less oriented a bit posteriorly. And I tell you that this is the area where prostate carcinomas are likely to take place, to actually find themselves here. Why? Because this area, the ducts of the prostate gland, open actually vertically, so that it can be that kind of retrograde of urine in here, and that is why this place is likely to suffer from prostatic cancers. Then what will happen is that mainly that is that that kind of reflux of urine into this place. You have to understand that. Then we also have what we call, so this is actually the area where, if during DRE, digital rectal examination, this is the area where the clinician in the gloved finger will actually feel. Okay, this is actually the area. It's more or less posteriorly related. Then we also have this one, which actually surrounds this prostatic urethra, so that if there's benign prostatic hyperplasia, it's actually this place where it's likely to take place, the transitional zone, and can compress on what we call the prostatic urethra. So that is actually going to occupy just about 5 to 10% of the functional volume of the urethra. Then there is this area which some people will call it the fourth zone of the functional zones of the the prostate, and it's known as the fibromuscular stroma. It's actually represent the front part of the prostate gland. So this place is less likely for either benign prostatic hyperplasia or carcinoma to take place. And it actually merges with the tissue of the urogenital diaphragm. The urogenital diaphragm will come this way, so it's able to merge with it. Now let me just tell you something about blood supply to the prostate gland. Yes, it will be supplied by prostatic arteries, which will actually is a branch of what we call the internal iliac artery, and also may receive some branches coming from what we call internal pudendal artery, as well as the middle rectal artery. Now for the venous drainage, they will be drained by what we call prostatic plexus of veins into actually what we call internal iliac veins. So one key thing over here, which is very important when you talk about carcinoma of the prostate, is that the prostate will have this kind of prostatic venous plexus, may have connection posteriorly to what we call the Basin's plexus, which is actually related in the anterior aspects of the bodies of the vertebrae, connecting to what we call the para vertebral what we call venous plexus. This connection represent a potential site for spread of cancer cells of the prostate. Okay, so that most spread of prostate cancer will get to the vertebrae first, okay, through this Basin plexus and into the para vertebral plexus of veins. So the main route of spread of prostate cancer is actually hematogenous means, through the blood. Now there's one thing which I'm going to show you, which we call it the cavernous plexus of nerves, which I will show you, which is very important for the autonomic innervation of the penis. Now if you have prostate cancer, which you have, therefore you have to take very good care of yourself so that you don't get prostate cancer. That's why you pray that you get it later in life by going through all those modifications of lifestyle. Because if you get prostate cancer, and there should be radical removal of the prostate gland, then what will happen is that there are some two nerves which I've told you, cavernous nerves on either side, related to either side of the prostate, okay, in the pelvic floor. Okay, because what will happen is that, yes, if you go through it, what will happen, there's likelihood that these nerves could be injured. Okay, so when they are injured, the likelihood that you will be impotent after the surgery of the prostate gland. So this is actually the histology of the prostate gland. There's the connective tissue stroma. Now within the connective tissue, there are muscles, okay, skeletal muscles in there, and that's why we describe it as being fibromuscular stroma. Then apart from that, yes, if you look at this, the epithelial liner is actually simple cuboidal. Although this one, yes, is somewhat, you can see maybe stratified, but it's actually simple cuboidal, as you can see in this case, simple cuboidal epithelium. But this one, this prostate gland is actually going to have, generally going to have compound alveolar gland. It's going to be a compound alveolar gland. But sometimes it could be compound tubular alveolar. So for instance, you can see this one is tubular in shape, alveolar in shape, or a sinus in shape, but these ones are tubular. So this one is actually compound tubular alveolar gland. Now, what we find over here is that within the lumen, okay, you can see there are some pinkish or eosinophilic structures over here, and these structures are known as corpora amnesia or prostatic concretions. So once we are seeing prostatic concretions, then it tells us that we are going to see is that of the prostate, because these are actually some remnants of glycoprotein materials, okay, coming from the prostate secretion over here. So if you have someone who is at the age of 60 who has ejaculated for quite a long time, you'll be seeing a lot of the prostatic concretions compared to someone who hasn't ejaculated before. So you may not see, any of these prostatic concretions. So that is that for you. Now this is actually the bulbourethral gland. Okay, bulbourethral gland. Now one thing is that yes, there's some kind of similarities between the bulbourethral gland as well as that of the the prostate gland. Yes, in that, yes, the bulbourethral gland will have some skeletal muscles, okay, in the stroma. So it will also be fibromuscular. Its stroma will also be fibromuscular. But what happens is that its epithelial tissue is supposed to be simple columnar, but usually this one we don't see. But you can see the epithelial line, the cytoplasm is columnar in nature, although this one we see it to be a bit cuboidal. Okay, so once you are seeing this, okay, then it tells you that you are seeing what we call the bulbourethral gland. That is the corpus gland. Okay, corpus gland. But sometimes, yes, well, you can see the cuboidal type actually, as we can see here, but generally it's supposed to be actually simple columnar epithelial tissue. Now, so those represent what we call the duct system. So it is left with actually the urethra, okay, to end with the duct system of the male reproductive system. Now what happens is that yes, if we take the penis will have its bulb, okay, actually anchored to what we call the pelvis, the pubis of the pelvis, through a ligament which we call a suspensory ligament of the penis. Now again, the penis will have true leg-like processes. These true leg-like processes are known as crura of the penis. And these crura of the penis, within it will have some cavernous or erectile structures. Those erectile structures are known as corpora cavernosa. There are two actually. So singular is corpus cavernosum. Corpus has to do with body. And then this corpus cavernosum is actually an erectile structure, so that upon arousal sexually, then what will happen is that blood will flow into it through the dorsal penile artery. I'm going to show you all these things, the deep penile artery, actually the cavernous arteries and all that. So what will happen is that yes, this one, the also surrounding the urethra at the underside of the penis will be another cavernous body, another erectile body, which you call it corpus spongiosum. Now this corpus spongiosum upon erection is not hard, it's a bit soft or weaker compared to that of the corpora cavernosa. So corpus spongiosum will lie underneath it, actually surrounding the urethra, okay, as you can see here. So that is that. Now the penis, apart from having a root, will have what we call shaft, or the body of the penis, the shaft or body of the penis, and then it will have what we call the glans penis. Now, on top of the glans penis, in an uncircumcised person, we are going to have, what we call the foreskin, or the prepuce. So that is what we are going to find with the glans penis. So actually, yes, within the glans penis, you may see the erectile tissues to here as well. Now look at the glans penis. There is this crown over here. After circumcision, you see this crown shaped area, and that is known as corona of the penis. Then when you look at the urethra, okay, urethra will have three parts. Actually, I will get to that one. We have the prostatic part, the part where it's surrounded by the prostate. We have the penile part, or the spongy part, where it's surrounded by the corpus spongiosum. And then we also are going to have the parts, we are going to have what we call the membranous part in the urogenital diaphragm. Okay, the membranous urethra, and then the spongy or the penile urethra. Now in the penile urethra, you can see this dilated area is known as fossa navicularis, actually fossa navicularis, or the navicular fossa, okay, over here. And then this is the external urethral orifice. So we are actually going to go through this. All right. Now remember that the male organ is for copulation is the penis. So the penis is the legitimate male organ for copulation. Even nowadays we have other illegitimate structures used for copulation, but the legitimate structure for copulation, okay, sexual intercourse between male and female, is the penis. Now within this penis, now this penis will be supplied by, is richly supplied, okay, because an erectile structure, for it to get erected, it has to be well supplied with blood. And by way of its blood supply, we are going to have it from what we call external pudendal artery, okay, supplying most of the skin over here. But importantly, we are going to have what we call the internal pudendal artery. Now this internal pudendal artery, yes, will actually give rise to what we call the common artery of the penis, the common penile artery, okay, common penile artery. Now this common penile artery will actually give off what we call the bulbar part, but in this case you can see what we call the bulbourethral part. It will give off the bulbar and of course the urethral part of it. Then even apart from that, we're going to have what we call this one known as the dorsal, okay, the dorsal penile artery will give off this branch which will run dorsally on the penis. So usually you see some blood vessels on the dorsal aspect of the penis, that is called dorsal penile artery. Then from the dorsal penile artery will give off some circumflex artery, okay. Then there is another artery which will come from the common penile artery from the internal pudendal, and that artery is known as the the cavernous artery, supplying blood actually to the cavernous corpus cavernosum, corpora cavernosa, actually giving off these ones. Then for the venous drainage, after the blood has gone in there after sexual intercourse, the blood will have to be drained through veins. If it persists, especially after 3 hours, especially in those with sickle cell anemia or any of the sickle cell diseases, or even if after taking in some of these drugs which is going to help you maintain erection for a long time, then what will happen is that there could be a lot of blood staying here, okay, and that's a painful experience, staying in here for about 3 or 4 hours or more, and that is what we call prapism. But it will be drained by what we call veins, okay, into the internal pudendal veins eventually. And the veins actually going to divide it mainly into 2 parts. We have that part which will drain actually the glans penis, and that part is going to be the dorsal artery of the penis. In any case, even the corona of the penis around here where the glans is, there could be other arteries like retrocoronal venous plexus, like the subtunical venous plexus. Then also, draining, the body area, you know, the main shaft area will be the circumflex, veins will be the circumflex veins. So the circumflex veins will drain actually the corpus, spongiosum, okay, corpus spongiosum, which is related where the urethra is. Yes. Apart from that, it also will drain part of the urethra. It will be drained by part of what you call the bulba. Part of it will be drained by the bulba, as well as, of course, the urethral veins. So the bulbourethral veins will drain parts of what we call the corpus spongiosum, and then, the corona area, getting to the glands area. Yes, it will be drained by the dorsal artery of the penis, but proximally part of it will be drained by what we call the circumflex veins. Now, usually the distal parts, okay, as you come more distally, as you know, what will happen is that the skin will be drained into what we call the superficial dorsal vein of the penis. Now, there's the deep vein of the penis, but the skin around here will be drained by the dorsal vein, superficial dorsal vein of the penis. But that one, when it drains it, it won't go into the internal pudendal vein, but it will go into what we call the savinous, either the long savinous or the small, actually the long savinous, vein. Long savinous, vein, or the great savinous vein. So that's the vasculature of, the penis, Now, for innervation of the penis, now the penis is receiving both autonomic innervation as well as somatic innervation. So the autonomic innervation will be coming from the inferior hypoglossic, plexus, plexus, which, combines. So inferior hypoglossic plexus will actually give what we call the sympathetic innervation. Then from the pelvis planktonic nerve, we will get what we call this parasympathetic innervation. Remember that parasympathetic is going to cause erection, and then sympathetic is going to cause, ejaculation. They will combine over here to form this plexus of nerve known as the cavernous plexus. Now, this cavernous plexus will therefore contain both sympathetic and parasympathetic fibers, okay, so that it can supply, what we call the erectile tissues of the penis. Cavernous plexus, supplying the erectile tissues of the penis, so that it can help in erection, okay, so that even after, ejaculation, there could be, what we call refractory period, so that the penis can, lie in a state of inactivity, and that is what we also call detumescence. Detumescence can also take place. Then there could also be what we call pudendal nerve from S2 to S4, nerve level. This pudendal nerve will actually, so I told you that this cavernous nerve is related where the prostate is. So if you go through radical prostatectomy, total prostatectomy, then what will happen is that there's likelihood that you may injure any of these, cavernous nerve. And when that happens, then impotence is what we are going to experience. So this is the pudendal nerve from S2 to S4. This pudendal nerve, yes, will also give some supply to what we call some muscles, which we call them bulbocavernosus and ischiocavernosus. This pudendal nerve, as I said, is going to be somatic in nature. This somatic innervation will be sensory as well as motor, so that the motor innervation is actually responsible for the sensation or the contractions that you've experienced, okay, actually perhaps during ejaculation in the bulbocavernosus, as well as, the ischiocavernosus muscles. When you ejaculate, you are going to experience all those involuntary contractions over here. Then one other thing too that you have to know is that, yes, this pudendal nerve, apart from supplying some innervation to these ones, okay, will also be important in giving some supply to what we call the inferior, to the rectum through the inferior rectal nerve, as well as, some perineal branches, as we've seen already. So that is it for you. So eventually this pudendal nerve will give rise to the dorsal nerve of the penis, actually dorsal nerve of the penis. Okay, so that is that for you. But let me just say this that the penis and any other male reproductive structure that we've seen will develop under the influence of testosterone only, under the influence of testosterone. Now remember that when you look at the glans penis, it will develop from what we call genital tubercle. Genital tubercle actually comes from the mesodermal portion of the genital tubercle, as well as the corpora cavernosa. Okay, the two corpora cavernosa. But the ectodermal portion will give rise to what we call the penile skin, okay, as well as the prepuce. Actually around 8 weeks of intrauterine life, it will begin happening over there. Now, one thing too that I have to tell you is that males are going to have this small orifice over here. Therefore, it's less likely for us to contract a lot of these sexually transmitted infections, unlike females, who are going to have larger opening. So, and of course males are usually going to give and females are the receiving end. So it tells you why females are going to be susceptible to suffering from most of these infections. Talking about lymphatic drainage, then what will happen is that they will actually drain into what we call the lymph node of Lockwood. That's the deep inguinal lymphatic nodes, deep inguinal lymphatic nodes, mainly through this area. All right, so that is that. But of course, sometimes we can drain into superficial and then into the deep inguinal lymph nodes. Now, I told you that now running through the penis will be the urethra. Now, the urethra in males is roughly 18 to 20 centimeters long, but in females, it's just about 3 to 4 centimeters long, and that is why females are even going to be prone to UTIs more than that of females. Now, what you see is that for the male, we are going to have the parts which lies in the prostate, and that part is known as prostatic urethra. The prostatic urethra, yes, will be having epithelial line just like the urothelium. So it will have what we call transitional epithelial tissue. Then in the membranous urethra, it will be pseudostratified columnar to stratified columnar epithelial tissue, and then that's around here in the genital diaphragm. Then beyond that, that's in the penile urethra, we are going to have pseudostratified columnar to stratified columnar, or stratified columnar to pseudostratified columnar, and then eventually getting to the orifice outside becomes stratified squamous non-keratinized epithelium. So that's the changes that the epithelial lining of the urethra actually receives. Now, in the prostatic urethra, actually the posterior as well as the prostatic urethra, that is where we have a lot of issues over there. And these issues, what I mean is that we are going to have what we call the urethral crests. Okay, so this is the urethral crest. This urethral crest will actually assist to ensure that there is no retrograde. Urinary bladder will be here, so that it helps prevent actually. The trigone is going to help do that. But in addition, the urethral crest over here will also help minimize retrograde of urine into the urinary bladder. Again, it's also going to help to actually minimize retrograde of semen during ejaculation, so that the semen will not go this way into the urinary bladder. Okay, so apart from the thickening of the lower aspects, okay, the apex of the urinary bladder, forming what we call the internal urethral sphincter, helping in that regard. The urethral crest is also going to help in that regard. Yes, apart from that, now you see that on the lateral aspect, okay, on the lateral aspect over here, we are going to find this area known as prostatic sinus. Now, within the prostatic sinus are some openings, okay, some ducts are opening into the prostatic urethra through this area, which we are calling it prostatic sinus, and these openings are openings of the various ducts of the prostate gland, okay, so that is that. Then we also have this area, okay. Now this area is known as colliculus seminalis, or the seminal colliculus, and it's also known as verumontanum, okay, verumontanum. So this area actually will surround this area where will actually surround the openings of what we call these two lateral, prostatic urethricle, as well as two lateral what we call ejaculatory ducts. Now there will be two ejaculatory ducts, and then within it we are going to have what we call the prostatic urethricle. And this prostatic urethricle, where is sandwiched between the openings of these two ejaculatory ducts, will be important in voiding of urine as well as in ejaculation. So I mean, these are the main things that we are going to see in the prostatic parts of the urethra. Now the histology actually showing a cross section, transverse section through the penis. The penis is going to have skin. Okay, so outer over here will be stratified squamous keratinized epithelial tissue. Okay, then we are going to have beneath that we will also have, okay, look at this, we will have what we call tunica albuginea. Okay, dense irregular connective tissue, okay, so covering it. Now this tunica albuginea will further divide it into two, so that at the end of the day we are going to have what we call the median septum, so that we can have these cavernous bodies being the corpora cavernosa. Okay, now where you have the urethra being there, yes, there will be tunica albuginea, all right, but the cavernous, the erectile tissue over there will be corpus spongiosum. So I told you that the corpus spongiosum will actually surround this urethra. Okay, that's what we are going to see. Just by telling you this, we don't have septum only for the penis, but we also have septum in the case of the scrotal sac. The left and right testicles will have to dwell in separate compartment, and they will be separated by what we call septum of, or the median septum of what we call the scrotum. But externally it will appear as a raphe, so the scrotal raphe will appear externally. So that is the the penis cross section through the penis. So actually when we look at skin, we actually show all those features that you see. So here we have the urethra, once again having this irregular stele shape appearance, okay, having the erectile bodies two around, okay, erectile structures being around, okay, and then you have the paraurethral glands over here, okay, which will actually drain into it. Okay, so this is actually the urethra. Now the urethra, yes, we also have inner longitudinal, middle circular, and an outer longitudinal. Yes, we already know that the urethra will be important, yes, for both bringing the ejaculate into the female, or for urination, but they do so at different times and not simultaneously. So a male cannot urinate into a female during sexual intercourse. Now there are some anomalies that are related to the penis, actually, and these anomalies, one of them is going to be what we call hypospadias. Now with hypospadias, instead of the urethral orifice, yes, opening only at the external urethral orifice, so that it comes outside, what happens is that it can open at any part underside the penis. Okay, so you can have this one, which we call the glandular hypospadia. Okay, just before you get to what we call the corona, okay, of the glans penis, you find that one there. Then it can happen in the penile shaft as well, being the penile hypospadias. Then it can even happen in the scrotal aspect, in the scrotum, getting to where we have the raphe. Okay, this is the scrotal raphe, and then within it we have the scrotal septum. Then this one is known as scrotal hypospadias. Okay, it's usually a congenital disorder, congenital abnormality. Then we can have epispadias. Now hypo means below, so that's below the shaft, or below the inferior aspect of the penis, or the ventral aspect of the penis. Now for the dorsal aspect of the penis, that's above it, we can have epispadias, and that epispadias can open usually around the dorsal aspect of the shaft of the penis. So these are some congenital anomalies that you may see. Now, there are certain, kind of disorders too, which is related to uncircumcised, scrotum. Before A3, if you are not circumcised, then you may be suffering from some of these things, which we call them phimosis and paraphimosis, because you don't get circumcision. Remember that circumcision is actually religiously accepted as well as scientifically proven to be very effective in ensuring that, yes, you don't harbor a lot of infectious agent in there. Now, usually what happens is that, yes, for such people, those who with uncircumcised penis prior to sexual intercourse, they have to retract the foreskin so that it becomes something like that, so that they can actually have sexual intercourse. Now, we are told that if you are having uncircumcised penis, then what will happen is that you are prone to suffering from a lot of these sexually transmitted infections. Now, for this one, for phimosis, what happens is that it's actually difficulty for you to retract the foreskin. Okay, difficult for you to retract the foreskin over here. Okay, and that is what we call phimosis. Now, for paraphimosis, you're able to actually retract it, but for you to actually bring it back to its anatomical place, then what happens is that it fails to come back, and that is paraphimosis. So it leads to some kind of swelling, around this area. That is called paraphimosis. Now, let me actually end with this by actually telling you that, yes, in fact, the sperm should be very well. In fact, the semen, the seminal fluid should be very well, so that it can lead to what we call fertility. For infertility, for most people, they may have that of a primary infertility, and others may suffer from what we call secondary infertility. Now, it is primary, it means that you've not, conceived at all before. Then when it is secondary, it means that you've conceived before, but you are trying to see if you can conceive again. Now, for the male infertility, then what will happen is that we have to investigate the sperm. We have to know if your testosterone level is adequate and all that. So let's actually get to understand that, yes, there's a normal sperm having its normal concentration, normal morphology, okay, normal motility. Then we have such a phenomenon, we call it normozoospermia. So if you hear of normozoospermia, then we are talking about a condition, in which you have normal morphology, normal concentration, normal, everything being normal, actually, in normal volume, actually, for, the sperm. But one thing that I have to even tell you is that we have something we call semen analysis. Now, after your ejaculate, we expect that normally you should have a minimum of 1.5, mils or centimeters cube. And then the pH, when we check, should be around 7.2 to about 8.0, which I've already indicated with the help of the prostatic secretions and all that. And then its liquefaction after ejaculation, we expect that maximum 40 minutes it should get liquefied. Okay, if it doesn't get liquefied, then what will happen is that in vitro situation, then in utero too, you may be seeing such a phenomenon, which means that the spermatozoa will be entrapped within the semen and will be able to fertilize the oocyte. Now, normally after ejaculation, we expect that you should have about 200 on average 250 millions of the spermatozoa. But if you don't have it at all, you should have a minimum of 20 million per male of the ejaculate, and then the sperm should be highly mitotic. Then they should have focus. They should be able to fertilize. They should move in a particular fashion. They shouldn't just be moving haphazardly, as you can see over here. This one, they are not having focus. They should actually have focus to fertilize an egg. So these ones, they are actually abnormal. We expect that, yes, there could be abnormalities in there. There could be abnormalities, but they shouldn't go beyond, more than 30%. There should be abnormalities, all right, but they shouldn't normally go beyond 30%. And what happens is that for the abnormality, the head could be absent, the tail could be absent, and they could be moving sluggishly. All these ones are abnormalities which you may actually see. Now, this is the normal morphology. If you're having such a phenomenon, we call it normozoospermia. Now, sometimes the volume, the quantity which we expect that it should be at least 20 million, could be actually go beyond less than 20 million, few of them, and that is what we call oligozoospermia. Now there could be a situation where yes, the movement could be impaired. If the movement alone is impaired, then we call it azetozoospermia. Sorry, asthenospermia. Then we can have a situation where movement is inadequate, morphology is inadequate, and that is what we call teratozoospermia. Okay, where morphology, most of them having their hairs taken out, having their tails torn, all these things, moving sluggishly. If I have less than 30% of your functional sperm doing that, then we call it teratozoospermia. Then we can have a situation where we have oligoasthenospermia. When we have oligoasthenospermia, then it means that yes, concentration is a problem, less than 20, motility is poor, morphology is poor. So if you have such a phenomenon, then we call it oligoasthenospermia. Now there are some people who will ejaculate, but their ejaculate will contain no spermatozoa, and we call such phenomenon azospermia. Azospermia. But sometimes some people may not ejaculate at all through whatever minutes of sexual intercourse. They don't ejaculate, and if they don't ejaculate, we call that condition aspermia. Or sometimes they may ejaculate, but it will be a retrograde ejaculate, so that it will get into actually the urinary bladder. It won't get into the female, and when that happens, then of course it may not lead to fertility. So these are some of the things that may lead to infertility. Of course, hormones may play a role, but regarding what we are doing as far as the male gamete is concerned, the sperm will have to be healthy enough so that they get fertilize the oocyte. Well, for females may their problems may be anovulatory cycle. Yes, it may be blocked uterine tubes. All these ones can actually play a role. It may be thick mucus over there. It may be antibodies, which of course the female is actually producing against the sperm. All these ones, it could be that yes, the female is suffering from a big fibroid which has actually occluded, where conception is going to take place. So these are some of the things which can actually take place, which may result in infertility. So I think later I will actually take my time to go through male and female infertility investigations. All right, this has been a very tall marathon. I hope you find it useful to your studies, and may God richly bless you.